Hi everyone, welcome back to my YouTube channel Smarted by Prashanthi ma'am. So today we are seeing the prokaryotic cell from the cell and basic unit of life, class 11 biology. Okay, this class is especially for Neet exam. So those who are preparing for the Neet exam, these videos are going to helpful for them. So do watch the video till the end. So we'll see the today's topic, prokaryotic cell. Okay, what is prokaryotic cell means? These are the primitive cells. Primitive cells means what? Oldest cells. Okay, these are the oldest cells which we found in the evolution process. Okay, so about the prokaryotic cell structure, everything we are going to see in today's video. So here prokaryotic cell itself, it is a prokaryotic cell. So these cells are very small in size. These cells are very small in size, but they grow very faster. So the prokaryotic cells are the oldest cells and they are the primitive cells, and they will grow very faster and they are very small in size. Okay, they are very small in size. Some bacteria, see example for prokaryotic cells, note down these examples. Okay, bacteria, blue-green algae, mycoplasma, pleuropneumococcal like organisms. So these all are the examples for the prokaryotic cells. So here prokaryotic cells, based on its shape, okay, based on its shape, the different types of prokaryotic cells are there. The organisms are divided into different groups. Okay, so for example, see shapes will be different. Some bacteria will be round shape, some bacteria rod shaped structures. Okay, some bacteria are rod shaped structures, and some bacteria are comma shaped structures. Some bacteria are spiral shaped. Okay, spiral shaped structures. So based on the shape of the organisms, these prokaryotic organisms are divided into different groups. We'll see one by one. See here, so the organisms are round in shape. The organisms which are round in shape, those organisms are comes under coccus. Okay, coccus. And then the organisms which are rod shaped, see here, rod shaped organisms are bacillus, like lactobacillus. Okay, so lactobacillus. Coccus means streptococci. Okay, streptococci. Vibrio cholera. Okay, Vibrio cholera. Cholera is caused by the organism is Vibrio cholerae. And then spirillum. See, spiral shaped organisms. So based on the shape, prokaryotic organisms or prokaryotic cells are divided into groups. Okay, one is coccus, bacillus. Write down these all points: coccus, bacillus, vibrio and spirillum. And what are the examples for prokaryotic cells? Write down this: bacteria, blue-green algae, mycoplasma, pleuropneumococcal, pneumococcal like organism, PPLO. Okay, PPLO. So those are the examples. And what is the main significant feature which is differentiating the prokaryotic and eukaryotic cell? That difference, that significant difference is naked DNA. So naked DNA is the significant difference between the eukaryotic and prokaryotic cells. Why? Because this DNA is nothing but genetic material. So the genetic material is not covered by any nuclear membrane. The nuclear membrane is absent in the prokaryotic cell. So here you have the diagram of prokaryotic cell. Observe this diagram. I am explaining in detail one by one. So here listen. So prokaryotic cells are covered by three membranes. See, normally the prokaryotic cell was covered by the cell envelope. Okay, prokaryotic cell is covered by the cell envelope. Okay, that cell envelope, it is not single envelope, it is made up of three layers. The cell envelope is made up of three layers. What are the three layers here? See, the outermost layer is glycocalyx. The outermost layer is glycocalyx, and the middle layer is cell wall. And the inner membrane, the innermost membrane is cell membrane. Okay, these are the three layers which together it forms as a cell envelope. Okay, so here see, small flow chart I made here. So cell envelope is divided, means it contains the 3 layers: glycocalyx, cell wall and cell membrane. So this glycocalyx, the outermost membrane, see the outermost membrane, it differs in the different bacteria. It differs in different bacteria means what? Some bacteria, it may contains the chemical composition as a slimy composition, means it contains maximum fluid content. Because of the presence of more fluid contents and chemical secretions over there, it is slimy. Slimy means sticky. Okay, the slimy, thin, smooth layer, glycocalyx in some bacteria. In some bacteria it may be tough, thick membrane. The glycocalyx, it may be thick and tough membrane, and it is useful for the protection of the organisms. So first outermost membrane is glycocalyx. Okay, glycocalyx, and the middle layer cell wall. Cell wall is made up of cellulose. Okay, cell wall is made up of cellulose, and this cell wall is responsible for protection of the damages and infections. So note down this point. What is the function of cell wall here? Cell wall is responsible for the protection of the cell from the damages and infections. So if any damage takes place, so that repair is going to be done by the cell wall. And then the third membrane is cell membrane, thin membrane. Okay, cell membrane is the thin membrane, and it is made up of lipids and proteins. Now see here, the nuclear membrane, see the fluid portion, the dots, everything, the fluid portion, the fluid matrix is known as cytoplasm. Okay, so the fluid portion present inside the cell is known as cytoplasm. Then what the cytoplasm contains? So what is the structures which is present in the cytoplasm, this fluid matrix? See here, it contains the genetic material. This is the genetic material. This entire portion is the genetic material. So this genetic material, if you observe here, there is no nuclear envelope. Normally, if you see the eukaryotic cell, it was clearly covered by the double membrane nuclear envelope. Double membranous nuclear membrane is present in the eukaryotes, but in the prokaryotes there is no membrane. Along with the genetic material DNA, so what is the genetic material here? It is a DNA, and along with that it contains a circular DNA. So it also contains the circular DNA. This circular DNA is also known as plasmid DNA. Okay, along with the genetic material DNA, it contains the plasmid DNA. Okay, it is a circular DNA. What is the function of plasmid DNA? See, plasmid DNA you will get in the genetics. Okay, so plasmid DNA, so in brief I am explaining this. Plasmid DNA is responsible for transformation. Transformation means what? So when it is transforming from one form to another form, especially when it is enters into the host body. For example, E. coli bacteria, when it enters into our body, it has to change its shape. For example, see here, so this is our genetic material. It is our genetic material. So when the E. coli bacteria, this DNA, plasmid DNA enters into human body, it has to combine with our genetic material as that particular host DNA. Completely, it is just similar to the host DNA. That transformation of the DNA is possible with the help of the plasmid DNA. So plasmid DNA is a small circular DNAs are present inside the bacterial cell. This plasmid DNAs are responsible for transformations, especially during the infections. During the infections, this bacteria which is causing diseases enters into the human body, and it is going to be transforms as a host DNA, then it is going to be increase its number. It is increasing its number with the help of this plasmid DNAs. Now Next one, the structures, mainly the cell inclusions. See, the cell membrane is going to be forming as the inclusions. This inclusion of the cell membrane is forming as the mesosomes. The structures which is formed by the cell membranes are mesosomes. Very, very important point, mesosomes. Mesosomes are formed from the inclusions of the cell membranes. Okay, enfolding materials, enfolding structures of the cell membrane are mesosomes. Along with that, so here prokaryotic cell does not contain, very important point here, the prokaryotic cell, if you see here also, there is no other cell organelles. There is no other cell organelles except ribosomes, except ribosome structures. Say except only ribosomes are present in the prokaryotic cell, and this ribosomes also attached with the mRNA. So these all are the ribosomal structures. These ribosomal structures all are attached with the mRNA. So that's why these ribosomes are known as polysomes. The ribosomes are known as polysomes. Why? Because here many ribosomes are present at one place, and together that structure is known as polysomes. And this mRNA ribosomes together, it is responsible for the formation of proteins. So protein synthesis takes place in the ribosomes only with the help of mRNA for messenger RNA. So the polysomes are helpful in the protein synthesis. Write this point also. Polysomes helpful in the protein synthesis. Next one. So these are the ribosomes. Next structures, see, some bacteria are motile, some are non-motile. Motile means they can able to move from one place to another place. Some bacteria, they can't able to move, they will stick to one place. Either they may stick to the rocks, other they may stick to the tissues, host tissues. It is made to stick to the host tissues. So what are the structures are responsible here for the movement of the organism? Okay, so those structures are cilia and flagella. So we know that locomotory structures are cilia and flagella. So here this prokaryotic bacteria contains the flagella like structure. See here, this is the flagella like structures. So in this flagella like structure, if you observe, it contains three parts. It contains mainly three parts. What are the three parts in the flagellum? Mainly it contains the tail portion, hook portion, and then basal body. See here I am writing flagella contains three parts. Flagella contains three parts. One is tail. So this one please, tail portion, hook portion, and then basal body. Okay, this basal body is connected with this cell envelope. Basal body is nothing but it is connected with the cell envelope. What is the cell envelope composition? Glycocalyx, cell wall, and cell membrane. Okay, and then hook is for the attachment, tail is for the movement of the flagellum. And along with this flagella, it also contains, observe this diagram, it also contains some long tubular structures. Long tubular structures are known as pili. And also it contains small bristle-like structures. Long tubular structures are pili. Small bristle-like structures are known as fimbriae. Pili and fimbriae. So this pili is connected with the cell wall and cell membrane. So pili or pilus also we can say, pilus is connected to the cell wall and cell membrane. Fimbriae, bristle-like structures. These bristle-like structures are responsible for, what is the function of fimbriae here? Fimbriae for attachment. So it attaches with either rocks or host tissues. Okay, Fimbriae, small bristle-like structures, okay, just like cilia, they are going to be useful for the organism. It may stick to one place, means like rocks. Already I said some bacteria are non-motile, so those non-motile bacteria can able to stick with the help of the Fimbriae structures. Okay, this is all about the prokaryotic cell bacteria structure once again. So quick recap I am going to do, listen carefully. So mainly prokaryotic cells are very small in size and they grow very faster. Example for prokaryotes, it will be bacteria, blue-green algae and PPLO, and then mycoplasma, smallest bacterium is mycoplasma. And structure, if you see, main difference between the prokaryote and eukaryotic cell is the naked DNA. Okay, there is no nuclear envelope around it, and the bacteria are different types of bacteria based on its shape. So the shapes will be spiral shaped and at the same time rod shaped, comma shaped, spirillum shape. Okay, so here there are different types of shapes. Based on that they are divided into coccus, bacillus, vibrio and spirillum. And then if you see the structure, the cell, prokaryotic cell is covered by the cell envelope. This cell envelope contains three membranes: glycocalyx, cell wall and cell membrane. Glycocalyx differs from bacteria to bacteria. Some bacteria may contains the smooth slimy layer and some bacteria may contains the tough thick layers for the protection from the damages and infections. And then cell wall for the protection, and then cell membrane is made up of lipids. Cell wall is made up of cellulose. And then inside, if you see, the fluid matrix is known as cytoplasm. Inside it contains the naked DNA, genetic material DNA. Along with the genetic material, long genetic material, it also contains the small spiral DNA that is known as plasmid DNA, which is responsible for transformation of DNA in the host cell. Next one, it contains the cell membrane enfoldings. Cell membrane enfoldings forms as a mesosomal structures, and along with the mesosomal structures it is also forms the vesicles, tubules, okay, lamellae. These all structures also enfoldings of the cell membrane only. Enfoldings are lamellae, vesicles and tubules, and these are responsible for secretion and absorption, respiration, increasing the surface area for absorption, everything. And then it also contains the ribosomes. All are connected to the mRNA, and this ribosomal structure together is known as polysomes. And along with that it also contains the locomotory structures like flagella and cilia. Along with that flagella, it is having the pilus fimbriae. So pilus is the long tubular structures, fimbriae are bristle-like structures for attachment. And along with that it contains the inclusion bodies. Inclusion bodies are nothing but it stores the reserve material. Food material is going to be stored in the form of inclusion bodies. So these inclusion bodies, for example, phosphate granules, phycin granules and also glycogen granules. So these all granules are going to store the food material. So these are the inclusion bodies. This is all about the prokaryotic cell. I hope you all understand the session. So I'll be back with another topic in the cell basic unit of life. All the cell organelles in detail we are going to see in our videos. Keep watching and please do subscribe and share, like the channel Smarted by Prashanthi.